and or I have told you that the temperature control will be present, and it will also find out the differential temperature powers, and also it will maintain the temperature within the two pans. And there will be a sample holder, which is a pans is completely made up of aluminum as well as the platinum. They will have a sensor which is made up of the thermocouple, which is made up of platinum, and it will also have a sensor, separate sensor heating for both the sample as well as. So you can find in the diagram for the both sample as well as the reference pan, we will have a platinum resistant heaters will be applied to it, or else a heat sink will be passed, so that the heat transfer will be occurs within the whole system. And this is mainly used for the phase transition, especially for the melting glass transition, and also for the exothermic decomposition studies. And also it will mainly this transition occurs because of the heat capacity, the heat differences which are being applied to the sample. There will be changes will occur from the one component to another component. For example, if you are using a protein, then a normal folded protein have been completely converted into an unfolded protein. That will be a denaturation occur. So how exactly the folding will happening for the protein studies and all, we can use this differential scanning color. Then we will talk about the curves, the overall heat flux as well as the temperature and time. We are going to plot a graph between the time as well as the temperature difference. So here there are possibilities of exothermal reaction and endothermal reaction. So we know that the endothermal is nothing but there will be absorption of heat from the surrounding. So releasing of heat is called as the exothermal heat. And here we are going to use two samples, like the sample 1 as well as the reference. We are going to heat it. So if it is attaining a melting point, then it will start showing a peak value. Either it may be a positive peak or it may be a negative peak based upon the sample which are keeping for this whole experiment. And this enthalpy change plays an important role here in DSC. That's why we have represented the enthalpy change in the enthalpy, which will gives the colorimetric constant as well as the area of the peak. So from the peak value, we can understand that the area is directly proportional to the heat observed by the whole reaction. So the height of the peak is also directly proportional to the rate of the reactions. So you can find in the two diagram. So this is the DSI curve. In this curve, you can find the amount of energy input have been observed, and it have reached to a high peak value. You can find a top one, which is called as the TM value, the melting point. And then once it have been reached the temperature, it will start releasing down the energy. So that will be, there will be change in the sample will start occurring, so that we will have a decline curve. So the bottom curve will initially have the baseline as well as the buffer variances. We can find one is for the protein, another one is for the reference. So there will be no any changes occurs in the reference, whereas in the protein denaturation, the almost 50% of the protein will be start degrading after it have been reached this melting point T. So what are the factors which is affecting this curve? The instrumental factors as well as the sample characteristics. So one is that Fern's heating rate, recording or chart speed, or the atmosphere or geometry of the sample holder, or location of the sensors, or sensitivity of the recording or recording systems, or the composition of the sample containers, or the amount of sample, or the nature of the sample as well as the sample patch. So these are the factors mainly observed. And also the particle size, heat of reaction, thermal conductivity and solubility of it. So you can find an amplifier at the end of it, so that from that we can observe the time as well as the differences in the time in both the reference as well as the sample value. The heat driven as well as the heat have been completely monitored by the automated system at a temperature which have been attached to the CPU. So So in the diagram you can also find out the same thing. Everything have been monitored automatically, so the programmer will be present. The amplifier can able to identify the temperature, and also the temperature will be controlled by automatically by the programmer, and we will have a recorded in each and every stages. So before applying heat and after a certain time, how exactly it have been reaching this melting point. Till that we will find out the differences by using one amplifier, and if it is reaching this melting point, we will observe through the average amplifier, and it will be monitored. So sample preparation. So we have to accurately write the sample, then we have to place it down in the pan which are present inside it. It is made up of aluminum, platinum or any nickel, and this pan will have opens. We have to find out any open or hole or any of the air have been affected this pan or not. And this material should be used for, if you are selecting aluminum for a sample as well as for reference, you have to use the same pan, and the bottom of the pan should be in a very good thermal contact so that the heat will be absorbed or sent down to the pan. And then the sample mass as well as the heat rate will also start increasing based upon the revolution. There will be a change in occurs after the run of your gun for a certain time. The sample shape, usually it should be in the uniform shape, and it should be in the pellet form, means then you have to cross section that, and the uniform spread of the sample is very important within the pan. The size is also important. If it is very small, then it will have very small sensitivity, and if the sample is very large, then it will increase almost the sensitivity of the devices. And it will also depends upon the size of the material we can able to measure this by using this DSC. And the extreme reactive nature of the sample is more important. Pure organic or for a pharmaceutical, we can go for 1 to 5 milligram, and for the polymers we can go for 10 milligram. Composite materials we can go for 15 to 20 minutes. So application of DSC, and it is for the protein stability and for protein folding, then for the liquid biopharmaceutical formulation, process development in protein engineering we will go for this, and rank order binding, antibody domain studies, and also for the characteristics of the membranes, lipids, nucleic acid, miscellaneous systems we will use this DSC. And for the assessment of the effectiveness of structural change on the molecular stability in order to measure the ultra light molecular interactions, and also for the biocomparability using during the manufacturing in industries. So these were the applications of DSC, and it also have some of the advantages, that is, it is label free, it can eliminate the dye, and the results are very reliable, and we can use a small volume is sufficient. It is easy to operate, and also it is automated for the high throughput. So mainly for this DSC is mainly used for the biochemical characterization studies and biocompatibility studies we are using it, and also for thermal stability, and also for the melting point screening, for optimizing the purification storage condition, and also to enhance the stability of a given product, either a protein or a biopharmaceutical products. We are using this DSC instrument. So that's all about differential scanning colorimetry. Thank you.